Fixation of brain means hardening of brain by specialized technique. So it could be studied more precisely. Silent features method, immersion fixation, perfused fixation, combination fixation. Perfusion fixation followed by immersion fixation, microwave fixation, electrochemical fixation. Advantages: better neuropathological examination. Disadvantages: immersion and perfusion fixation takes 4 weeks. Study is delayed. In medico-legal examination, such a delay is generally not permissible. Microwave fixation is quicker but may affect subsequent histochemistry and immunohistochemistry steps. Immersion fixation. Brain is suspended upside down in a large bucket of 10% formalin for at least 4 to 6 weeks. Higher concentration make the brain very brittle and difficult to handle. Volume of formalin in bucket at least 10 times the volume of the brain, about 15 ml. In fetuses and infant, acetic acid may be added to formalin, thus makes the brain firmer. Add kosher salt. This floats the brain, minimizing artifact produced by compression of the soft, unmyelinated brain tissue against the walls of the vessel. Percutaneously inject fixative into the lateral ventricle. Advantages: preserves delicate pathological anatomy in cases of hydrocephalus or a poor encephalic system. Useful in situation in which special diagnostic studies, electron microscopy, in situ hybridization are required. Suspension method: upside down. A paper clip or curtain hook is placed under the basilar artery and is hung upside down by a length of string tied between the two handles of the bucket. Straight, less commonly used. Dura is used as an anchoring point. Parenchyma is deformed more by this method. To facilitate penetration of formalin, lateral fissures are opened with the fingers. This tear opened the arachnode following penetration. If fixation is likely to be impaired by edema, corpus callosum can be split to access the ventricle and thus improves the penetration of the fixative. Layers of cottons are kept at the bottom of the bucket so the top surface of brain does not get flattened against the bottom of bucket. Gauze should not be used as it leaves its imprints. Several changes of formalin should be made, preferably after 3 days initially and then weekly. Weight changes causes about 10% of weight brain in adult and 15% weight of brain in children. Perfusion fixation. Brain is perfused with fixatives under pressure through vertebral and internal carotid artery. Can be done when brain is within the skull or after taking it out. Advantages: somewhat faster than immersion fixation, especially for deep-seated regions of the brain. Improves immunohistochemical staining. Disadvantages: artifacts are noted. Irregular weight matter, pallor seen by hematoxylin and eosin stain. Simulates white matter diseases, but there is no attendant gliosis to signify antemortem injury. Vascular disease, atherosclerosis or injuries, inadvertent laceration of the circle of Willis. During brain removal, make perfusion difficult or even impossible. Thus, in case of suspected cerebral embolia or thrombi, perfusion fixation of the brain is contraindicated. Microwave fixation. Thank you.